Right, today's lesson is super short. I missed it somehow. So this lesson goes before translation. So this lesson is about how DNA is transcribed and we get a single RNA transcript. So there's a few different steps, generally 3 steps. One is initiation. So you have there's only one enzyme involved here, which is RNA polymerase. It binds to the DNA and unwinds it. It binds at a promoter region called the Tata box, and RNA polymerase recognizes this as a start sequence. Now, the Tata box, the reason that we have the TATA combination is that A and T bond only 2 times, so it takes less energy to start at this start sequence. So here's a picture. Here's the RNA polymerase molecule. Here's the DNA being unwinded by the polymerase. It's not shown here, but it starts at the Tata box unwinding. Okay, so the next step is elongation. So you can see there's no lagging there's no lagging and leading strands here. Here's the DNA template. You can see the nucleotides coming in here, and the one difference is that here we have the uracil instead of thymine. This part is termination. So the RNA copy is being produced continuously. When it gets to the end of the gene, it drops off. You get this RNA here, and the DNA rewinds. One difference here between DNA replication and transcription are the post-transcriptional modifications. So we have the DNA template here. You have the promoter region with the Tata box. We have the coding region, non-coding region, coding region, non-coding region, coding region. So we have the 7 guanines here, which is sort of like a start sequence for the ribosome. And we have a poly A tail here, and then these 2 are cut out, spliced out with the introns removed. Okay, so this process is called capping and tailing. These are considered post-transcription modification. As the mRNA goes out into the cytoplasm, it's vulnerable to enzymes, which the DNA isn't because it's in the nucleus. So to protect it, it has to undergo these modifications. So the addition of the 5' cap, that's the initial attachment site, and the addition of the poly A tail, it protects it from being attacked by RNA digesting enzymes, and it enables RNA to be translated efficiently. So the addition of this and this. Okay, a little more detail about removing the exons from the mRNA. The DNA is made up again of the introns, which are the coding regions, and exons, which are the non-coding regions, and they are taken out or excised. So here is a coding region and a non-coding region and a coding region. We have these proteins, and this cluster together are called, I think it's pronounced SNRPs. They attach to the intron, which is a non-coding region, and they cleave it, and the intron folds back into itself, and then it's released, and then these 2 segments on either side are attached together. So just a closer look. This complex of the SNRPs plus the non-coding region together is called a spliceosome. And that's it. Shortest lesson ever.